Welcome back to my YouTube channel. So today we are starting a series of what we call the pre-fertilization events. Now this will form the basis for understanding human sexual reproduction. So here we'll talk about how the gametes are formed and what happens during fertilization. And so before we go into all that, we'll start with the concept of chromosomes. Now this is a normal, single, unduplicated chromosome. You can be able to see that later, one chromatid will duplicate and form a sister chromatid. Now, each chromatid is divided into a short arm, what we call the P arm, and a long arm, what we call the Q arm. And they are joined at the centromere. So this is what we call a duplicated chromosome. So what you form here is a duplicated chromosome, and this is an unduplicated chromosome. Now, this tail end we'll have what we call the telomeres, and we shall discuss that in our next video. There is a special term in terms of chromosomes, what we call ploidy. Ploidy refers to the number of chromosomes within a cell. So when we talk about diploid, what that means is you have 46 single chromosomes. Now haploid means that you have 23 single chromosomes. The capital N is actually a number that refers to the amount of DNA within a cell. So normal somatic cells, that is the epithelial cells, the connective tissue cells, they will have 46 single chromosomes and 2n the amount of DNA. So these 46 single chromosomes will be paired into what we call homologous pairs. So each pair will contain a contribution from the mother and from the father. So we say that within the 23 pairs, you have a contribution for each pair, you have a contribution from the mother and from the father. Now for the gametes, what usually happens is you only have one sex chromosome. So usually you just have a single X chromosome for the ovum, and from the spermatozoa you have either an X or a Y chromosome. And now this is very important because this means that the male gamete is what determines the genetic sex of an individual. And the key to this sex determination relies on the presence of what we call the SRY gene. The SRY refers to the sex determining region of the Y gene. Now when it's present, you have what we call differentiation of the different gonad to form the testis. When it's absent, the default status is to form the ovary. And so this is what we call the SRY gene. It's part of the short arm of the Y chromosome. And so this SRY gene produces what we call the testis determining factor, and this is what promotes the formation of testosterone in the embryo and allows the differentiation into the testis and the phallic structures. Now, gametogenesis is a process whereby you form and develop gametes. And when we talk about gametes, we're talking about the spermatozoa and the ovum. And so the formation of the sperms is called spermatogenesis. Formation of the ovum is called oogenesis. And both occur within their respective gonads, that is the testis and the ovary. So we'll have an individual video on spermatogenesis and another video on oogenesis so that we can be able to look at them in depth. So in the developing embryo, what usually happens is the indifferent gonad starts to form, and then primordial germ cells come from the yolk sac and go into the developing gonad. Now these primordial germ cells will then differentiate to give stem cells, which can either form sperms or ova. Now the primordial germ cells will have 46 single chromosomes and 2n the amount of DNA. So this is a summary of the process of meiosis. The key thing to remember is that what you usually have is 46 homologous chromosomes. And when you say you have duplicated chromosomes, remember this is what we mean. That this is a duplicated chromosome, this is a duplicated chromosome. This is a single chromosome, this is a single chromosome. And so you usually have this pairing of 46 homologous duplicated chromosomes. And then at the level of the chiasma, there is usually this crossover of genetic material. And this is what promotes genetic variability once fertilization occurs. So the splitting of the cell will be able to send each homologous pair into its own cell when you have what we call separation or disjunction. Now remember, at this stage of meiosis 1, there is no splitting of the centromere. Now when you go to meiosis 2, the secondary gametes will start to align at the metaphase plate. Now, when they align at the metaphase plate, this now prepares them for splitting, and they split at the level of the centromere. Now, once this split occurs, you're able to form 4 gametes, each with a single unduplicated chromosome, like this. Okay, and so they will have 1n, the amount of DNA. And so this is a summary of the process of meiosis. Now, this is how normal meiosis should happen. Pair of homologous chromosomes, okay, they meet at the metaphase plate. Then from the metaphase plate, you usually have the splitting of the cell. And then now from here, what you have is splitting at the level of the centromere that gives you a single unduplicated chromosome in each cell. So from here, you get 2 gametes. Now from here, you should also get 2 unduplicated gametes. Now, unfortunately, during meiosis 2, there was no disjunction. There was no separation. Okay, so this should have been here, but now there was no disjunction. And so this usually leads to problems like Down syndrome, Edwards syndrome, what we call the trisomies, and we will discuss them further on the video on fertilization and its results. So thank you very much. If there are any questions, you can leave them on the comment section below.